Shortly after uploading my video where I criticized common descent by demonstrating conflicting homologies between bony fishes and cartilage fishes, one of my favorite evolutionist critic, Mr. alMind, came to the defense. In his stance, he proposed that even though there are homologies present in otherwise two different taxon groups, one of that being closely related and distantly related to the tetrapods, the bony fishes, and the contradicts, the cartilage fishes, he proposed that it does not contradict common descent. What was the defense? Whole genome duplication, as alMind quoted from the same paper. Basically, what alMind and the scientists in the paper try to point out was that the reason why the bony fish groups possess less genetic similarity or synteny compared to that of distant groups like the cartilage fishes, such as the elephant shark, was because there was a whole genome duplication event that occurred during the fishy bone lineage. At the same time, the whole genome duplication has caused drastic changes in the genome of bony fishes, such as large-scale rearrangements and high rate of gene deletions, which eventually evolved as lineage-specific genes, a process called gene fractionation. Therefore, under this very reasoning, it is more likely that the genome of the bony fishes have underwent massive deletions, inversions, and genomic rearrangements, explaining away why tetrapod vertebrates like humans and mice share more strong genetic similarity with that of non-bony fishes, such as the shark. While this may seem as a reasonable constructive criticism against my argument criticizing common descent, there are actually flaws with the whole genome duplication model. Say hello to the Rainbow Trout entitled in the 2014 paper. The Rainbow Trout genome provides novel insights into evolution after whole genome duplication in vertebrates. Here, researchers examine a recent whole gene duplication event named the alMind specific fourth whole genome duplication, otherwise abbreviated as SS4R, that has been dated 25 to 100 million years ago. Due to the recent SS4R arrival, researchers had the opportunity to better understand the early steps of gene or otherwise whole genome duplication. They performed an analysis on the whole genome sequence of the Rainbow Trout. By examining the SS4R regions, they discovered that most of the duplicated regions have revealed a surprising discovery. They found that nearly all of the duplicated regions have remained highly conserved and hardly diverged for 100 million years after the SS4R event. Furthermore, they found that despite 100 million years of evolution after the SS4R whole genome duplication event, they found that the ancestral gene and gene copies of it have extremely stability, emphasizing that the duplicated regions have remained remarkably well conserved in sequence identity and gene order on chromosomes, which demonstrates interesting crucial points against the whole genome duplication hypothesis. 1. Whole genome duplication does not involve many genomic rearrangements such as inversion or translocations that will otherwise modify the order of genes in the genome. 2. It hardly involves deletions and neo-functionalizations. And 3. Even among pseudogenes caused by failed gene duplications, they remain highly strongly conserved to that of the functional protein coding gene copies. This is quite contrary to what alMind and the scientists in the paper emphasize, that the absence of closely related genes can be explained by whole genome duplication events, given that they involve large-scale genomic rearrangements, deleterious mutations, and neo-functionalization. Rather, what this study reveals is that the rate of deletion, rearrangements, inactivation, and so on are extremely slow and rare. It is in fact so slow and rare that even after 100 million years after the SS4R event, there still remains a very high rate of conservation in the duplicated regions, suggesting the extreme rarity of gene duplication, deletion, in the 100 million year time scale. Thus, whole genome duplication is not a valid answer for the lack of homologous genes in the bony fish lineage with that of the tetrapod vertebrates. So unfortunately for alMind and the scientists, they have yet to find a reasonable explanation for why the bony fishes are missing such genes in the defense of common descent, given that whole genome duplication is extremely rare and hardly practical in the real world. Thanks for watching.